So hello, and much welcome to the first EMBO ECR online panel discussion of 2023. My name is Andreas, and I have the honor to co-share this session with my two fellow ECR colleagues, Said and Ersin. And in today's session, we'll explore non-academic career pathways. And to provide some insight on this, we are happy to be joined by Chris Rose from Genentech, Lindsay Pino from Talus Bio, and Maria Polychronidou from Molecular Systems Biology. And with that, I will hand over to Ersin.
Hi, everyone. So I have the pleasure of introducing Dr. Rose. He's a senior principal scientist and director of the microchemistry, lipidomics, and proteomics department at Genentech. He completed his PhD in the Kuhn lab at the University of Wisconsin-Madison, and then went on to do a postdoc in the Gigi lab at Harvard Medical School. His current research focuses on two main areas, where he looks at real-time searches for low input and single-cell samples, and the application of MHC-1 into peptide analysis to explore processing and presentation of cancer-specific antigens in tumors. He's also a postdoc mentor at Genentech and collaborates with many ECRs in various capacities. We look forward to hearing about his career path thus far and his takes on non-academic career paths in general. Over to you, Dr. Rose.
Great, awesome. Can you hear me?
Yep.
Great. All right, let me also share my screen. All right, let me know when you can see the slides as well.
Yep, it's coming through.
Yep, you can see it. Okay, great. Yeah, I can make this kind of brief. So I'll give you a little bit about my background and then also how I made it to Genentech. So I'm actually from California originally. I grew up in the Bay Area and I went to college at a smaller liberal arts college called Santa Clara University, maybe most famous for being where Steve Nash went to school. But we've also had some other people as well. And there I studied both biochemistry, which was in the chemistry department, as well as cellular and molecular biology, and did some undergraduate research on capillary electrophoresis selection of aptamers. And that got me really interested in kind of the research path. So from there, I looked at a lot of different opportunities really within the bioanalytical space, kind of mixing both biology as well as the analysis of those samples by instrumentational means. And that kind of landed me in Josh Kuhn's lab. And in Josh Kuhn's lab, kind of right from the start, I worked on a lot of multiplex quantitative proteomics. And also in that space, I also did some work on some of the very beginning of the real-time data analysis strategies. But also while I was there, and this is kind of one of the key points to my journey, I actually did a collaboration with the department I'm currently in at Genentech. So I was still a graduate student, and the department I was in actually reached out to Josh because we had a technology that we thought was going to be interesting, and we were kind of looking for a biological sample. So we linked up with the current department and Don Kirkpatrick specifically, and we did an experiment together with one of the postdocs in the lab. And that got me kind of really aware of what Genentech was doing and the kind of research that was happening here. From there, I went to Steve Gigi's lab at Harvard Medical School, and I kind of continued that theme of both multiplex quantitative proteomics as well as implementing kind of these real-time search strategies. That was particularly with Tomahawk, but I also wrote some of the proof of concept work for the real-time search that then later was finished off by some other folks in the Gigi lab once I left to go to Genentech. So after about a couple of years in the Gigi lab, I kind of had a choice to make. I was thinking about, whether or not I wanted to try to go more towards, doing more publications, maybe trying to get a professor job somewhere. But at the same time, a job opened up back in California in the current department that I'm in now. And I decided to go ahead and apply for it, mainly because of the experience I'd had with Genentech, as well as kind of the reputation with the group. And since then, I have a number of different responsibilities. So I started at Genentech as what would now be a principal scientist. The titles have changed quite a bit, but it would now be a principal scientist that's a group leader. At the beginning, for the first year and a half, I actually didn't have anyone in my group, but my focus was really on kind of developing and modernizing a lot of the mass spec approaches that we had in the group. In addition to that, doing a lot of collaborative research. So the job initially, right, was a lot of work in the lab, not only setting up the mass specs, but also working with collaborators in all the different therapeutic areas around Genentech on the types of samples that they were interested in getting biological questions answered. In addition to that, it's been a lot of technology development. That's obviously methods on the sample preparation side, but also on the instrument side, specifically around that idea of real-time search and some of the software that we've developed internally to do things like the real-time search. And in addition to that, another portion of my time is really spent on computational proteomics, where I've actually helped people analyze data, written some algorithms to kind of push data through and whatnot. So currently, this is where I'll spend another minute or so. But currently, my position is really the director of discovery proteomics within the microchemistry, proteomics, lipidomics, and next generation sequencing department. It's the longest name, I think, maybe in the company. So we have both NGS and proteomics in the same bundle, which is great. My particular group, part of the department, focuses on discovery proteomics, which traditionally is a lot of the bottom-up proteomics. And that focus is really to work with the collaborators in different therapeutic areas as they come up with questions about basic biology, typically on things like proteins or pathways that might be future therapeutic targets. We typically work with people very early on when it's really just a hypothesis, all the way through when they have, say, a molecule and they're trying to understand what that's doing in a cell. So we take it almost all the way until they already have kind of validated the molecules, validated the targets, then it gets handed off. And within the discovery proteomics group, my group is just one of them, really focusing on low input samples and advanced instrument control. But we also have a number of different group leaders focusing on various areas, from immunoproteomics, single cell molecule sequencing with Susan's group, single cell and spatial proteomics with Ying, and then Hannah has been focusing on chemoproteomics. So we have a very diverse department, and that's part of my role now, is to kind of oversee the direction of the department, the resourcing for the department, and other things like that. So with that, I can hand it over who's ever next.
Thank you so much, Dr. Rose. I guess it's Saeed next.
Yes. So I have the pleasure of introducing Dr. Lindsay Pino. So Dr. Lindsay Pino graduated from the University of Washington in July 2019 with a PhD in genomic studies, where she develops approaches for large-scale quantitative proteomics using data-independent acquisition mass spectrometry. She is the co-founder and chief technology officer at Talus Bio, and with over a decade's experience in analytical chemistry and computational biology, she has been developing technologies for quantitative proteomics. At her current place, which is Talus, she works on the challenges associated with scaling up quantitative proteomic experiments, and is directly involved in a large variety of research projects spanning cancer drug discovery, molecular mechanisms of aging, epigenetics, and spatial proteomics. So with that said, Lindsay, if you could share your slide, that would be great.
Great. Does this look good to everyone?
Yes.
Awesome.
You're good to go.
Yeah, so I'll start out by saying my story is maybe a little weird in that I started off in undergrad wanting to study biochemistry and molecular biology because I was just very inspired by this little kid, Maddie Stepanek, who had a very rare but fatal disease called mitochondrial myopathy. And we knew exactly what caused mitochondrial myopathy. We knew exactly what potential cures would be, but we just couldn't do it. It just blew my mind that we could know the cause of a disease and we couldn't fix it. We didn't have enough people working on solutions to cure these diseases. So I was like, I'm going to be one of those people. And clearly, as you can see on the left, I burnt out pretty fast, not really quite making it. In science, I decided I was going to take a couple of years and I moved to South Korea, where I taught English for TOEFL students. And I really loved teaching. It was a lot of fun. I really thought I was a great teacher, but I missed science, and science is not something you can kind of do as a hobby. You can teach on the side. You can be a scientist and teach. You can't really like be a teacher and do R&D, research scientist. So I came back to the States and I decided science is my thing. And I started out back in the States in a genomics lab. And within a couple months, I realized genomics is not my thing. Genomics is great. And there's a lot of cool things we could do with genomics, but not for me personally. That's not where my passion was. So I ended up in the proteomics platform at the Broad Institute, where I really kind of hit my stride. This was I loved doing what I was doing. I was working with mass spectrometers. I was working with hardware. I was doing proteomics. I loved it, despite still making many mistakes, like crashing a $100,000 robot head and costing the lab quite a bit of money in my efforts to do technology development. But regardless, I had found that proteomics was definitely my thing. This was something that I wanted to do forever. And that's when I finally decided I was going to go to graduate school. But even graduate school did not really progress linearly. So I started graduate school at University of Washington in the Genome Sciences department. And before I even started, my first thesis proposal was rejected. And then I started and my next thesis proposal was rejected. And then my third thesis proposal was also rejected. So clearly I was kind of still figuring out what it meant to be a scientist and do science. So finally, my fourth research proposal was accepted, and that was great. I did my first set of projects, which involved calibration curves for mass spectrometry. I really loved this kind of work, this kind of analytical chemistry style of work that merged doing some sample preparation, but also working on an instrument and kind of like the hardware and methods development on a mass spectrometer, plus doing the computational side and coming up with new ways of analyzing data. So now, finally, like, man, it was like 6 years after I finished undergrad, I finally figured out what I was doing and what I wanted to do for the rest of my life. And then once I hit my stride, then things started kind of stepping up. I won a graduate student award. I got the first paper out. I started a second project. I got a second paper out. Things were starting to like hit a groove, and proteomics definitely felt like it was my thing. I was giving talks at ASMS. And in the course of kind of carving out this niche for myself of doing quantitative proteomics, I was starting to pick up collaborations with other people in my department, in my PhD lab, who were doing cool things but needed help with their mass spectrometry or their data analysis or ways to prepare their samples. One of these collaborations was this cell-based discovery of transcription factor inhibitors. So kind of the goal of this project was my collaborator and now my co-founder, Alex Federation, had this approach where he would take cells, take nuclei, he would wash out the transcription factors and then measure them by mass spectrometry. So you would get a read of which transcription factors were bound to DNA and which ones were not bound to DNA. And through measuring those, he wanted to discover these small molecule inhibitors of transcription factors. So this was like the perfect kind of collaboration where you have a coworker or a collaborator where you think their science is cool, they think your science is cool. You can work for 12 hours a day with them and then still go out and like grab a beer and dinner and not feel like you're overwhelmed and like, I just need to go home and get a break from you. This was like, loved working with him. So when he reached out and wanted to start a company, I was like, sure. And it turns out there's a lot of unmet medical needs with transcription factors. The drugs that are out there were kind of just discovered by serendipity. And of the hundreds of transcription factors implicated in cancer, there's only 18 approved drugs for them. So there was a lot of space for us as a very small company to start working. So that was really kind of cool. And when he proposed this idea to me of starting a company, we kind of had this weird moment of like, what even do job titles mean? So like Chris had kind of mentioned how job titles change rapidly over time. Job titles are also like basically meaningless in a small startup company. So he was like, what do you want to call yourself? I was like, I guess I'm a CTO, because of all the things that we're doing at the company. We're doing mass spectrometry proteomics. We're doing kind of sample prep for cancer biology. And we're also doing AI data science work for analyzing the data. And I'm involved in all of these pieces, these rapidly moving and changing pieces in different parts of the company. So I thought a technology umbrella would work really well. I work with a very small team. This is literally everyone in the company. It's about the size of maybe like a medium to large academic lab. But I work really closely with each one of these people in the various roles that they play within the company. And an average day for me kind of changes over time, but if I were to take a running average, I spend almost half my time in meetings. I spend on average about 10% of my time writing grants or doing fundraising. about 10% of my time developing the business and making sure the company kind of like is known in the pharmaceutical and in the proteomics space. About 20% of my time is just administration and management work, and only 20% of my time I actually end up doing the science. So I'd be happy to answer any questions that people have about any parts of that when we get to the Q&A.
Thank you so much. I will give that to Andreas for our final panelist.
Yeah, so I'm happy to introduce Maria Polychronidou, who is a senior scientific editor at Molecular Systems Biology. She received her PhD from University of Heidelberg, where she studied the role of nuclear membrane proteins in development and aging. And then during her postdoctoral work, she focused on the analysis of tissue-specific regulatory functions of Hox transcript factors using a combination of computational and genome-wide methods. And then in 2013, she joined Molecular Systems Biology. So I'll hand over to Maria.
Thank you. Let me start sharing my screen. Let me know if that works. Can you see my screen?
That works
Yeah.
perfectly.
Okay, great. So yeah, as you heard, I'm senior editor at Molecular Systems Biology. This is one of the 5 journals that are published by EMBO Press. The rest is shown on the bottom of this slide. Maybe you have read papers in one or more of them. And for today, I'm going to focus on my role as an editor. But in addition to that, I have a second role, which is that of a training coordinator at EMBO Press. So briefly to mention what that is about. There is some scientific skills training that has been developed by editors at EMBO on the topics that you see here. So one of the courses is about scientific integrity, so basically how to conduct research in a responsible way. Another workshop which I developed together with a colleague of mine deals with how to communicate research effectively in a written or oral form. And then there's another module, how to review a scientific paper and how to apply design principles to schematic figures. And part of my role is to coordinate the delivery and the development of all these courses. And if that's something that any of you are interested in, you can find more information on the website that is shown on the slide. Just a little bit before I go into what my role as an editor is about, just a brief summary of how I ended up there. So I did my diploma in biology at the University of Athens in Greece. As you may have guessed from my long name, I come from Greece. Then I moved to McGill University in Canada, where I did a master's in biochemistry. And then I moved back to Europe. I went to Germany, to the University of Heidelberg, where I did my PhD, and later on a very short postdoc that was actually only for 2 years. So I realized fairly early on, actually already from the end of my PhD, that I most likely would not want to pursue a career in academic science. But I nevertheless decided to do a short postdoc. And then in 2013, I was hired as a scientific editor at MSP. And in 2019, I also started my second role that I just described as a training coordinator. So how did I ended up becoming an editor? As I said, I realized that the academic career path was not for me. This was not something that I really could see myself enjoying long term. There were certain things I liked. So I really liked reading papers. I liked writing papers and grants. I really enjoyed presenting papers that were out of my comfort zone. So I was always picking, whenever there were journal clubs, I was picking papers on topics that really had nothing to do with what we were working on in the lab or even in the small department where I did my PhD. And I really always enjoyed learning new things. And as I said, I realized very early on that one of the things I really wanted to do was leave the bench. But what I did not want to do is, I did not want to leave science. So taking into consideration all that, I decided to explore some alternative career options, and in the end, I ended up applying for editor jobs, and I was hired as an editor at MSP. So, how does a typical day look like in my job? So, the main chunk of the time is devoted to evaluating the manuscripts that are submitted to the journal. So, typically, I read, and it's the same for the other editors in the team. I read something like 3 manuscripts a day. I decide if they are well suited for the journal or not. This we do as a team. If we decide to send these manuscripts out, then one of the tasks is to find referees. And then once we receive the referee reports, we have to assess them and make decisions whether we are going to invite review or if we are not going to proceed with publishing that specific paper. Another good chunk of the time goes to writing lots of emails, communicating with authors, reviewers. Sometimes we have calls with authors to discuss different things, like, for example, appeals on decisions. If the authors do not agree with the decision that we make, they can always appeal, and we can discuss that in a call to get more information. And we also provide advice on revisions, if the authors are not so clear as to how to address the issues that were raised by the referees and things like that. And of course, we also write these emails and have calls about other inquiries. And finally, another thing that we do as editors is to commission review articles, but also news and views that highlight papers that we publish in the journal, and commentaries and other pieces for what we call the front end of the journal that is not related to primary research. Some other things that we do include occasionally having strategic meetings with the team and the editorial advisory board of the journal. And this we do to make decisions on strategically developing the journal further, defining the scope and things like that. We also attend a lot of scientific conferences, but also other events, for example, institute retreats or courses or career events like this one. And we also, we visit labs and institutes. The purpose of this is to get to know people and to allow them to present to us their research while it is still unpublished, and potentially explore the possibility of submission to our journal. Before I close, just to mention some things that I personally enjoy about the job. The first one is that you really get to know as an editor what is new and upcoming, because you see everything while it is still unpublished. So this is really quite exciting. You see the research while it is being shaped before it is published. You get to know a little bit on a lot of different topics. So of course you cannot be an expert on the topics like the researchers that are working on these topics, but you get to know a little bit about it, and you are really always learning something new, which I personally find very exciting and intellectually stimulating. And as I mentioned, we're also attending quite some conferences, which is really nice. We meet a lot of people, and it really feels like you're part of a scientific community, which again, I personally enjoy very much. And finally, we get to work with the authors to get the study published. So it always gives a good feeling in the end when the paper is published and the authors are satisfied and they tell us that they were happy to work with us. This is always very, very fulfilling. But of course, on the other side, there's some aspects of the job that maybe are not so nice. The first of them being that we unfortunately have to communicate bad news very frequently. We reject quite a lot of papers. This occasionally, not very frequently, but occasionally it can lead to bad reactions. So this is not particularly pleasant. Something else to keep in mind as a downside, if you're considering this as a career option, is that we have to work under tight deadlines. So imagine, I guess all of you have been in a rush to submit and publish your paper because you have a grant deadline or because you have to graduate and so on. So imagine that multiplied by the number of papers that each editor has to handle. That means really working under tight deadlines for a large number of manuscripts. And as exciting as it is to travel and go to conferences, the traveling schedule can get a bit heavy sometimes. That's maybe also something to keep in mind. And finally, something that for me personally has not been a problem, but I know colleagues of mine have find it as a downside of the job, is that while you get to follow many exciting developments, you do not make discoveries yourself anymore. So you get the excitement about science via the discoveries of other people. So again, that's something to keep in mind. So with that, I'm going to stop. I hope I gave you a flavor of what the job is about, and I'm happy to take questions.
Thank you so much, Maria, and a massive thank you to all the speakers who have had amazing presentations so far. So I just want to point out to all of the attendees that you're more than welcome to post questions in the Q&A tab if you have any questions for the panelists. And to open things up, we have prepared some questions that I thought a good way to start is to ask them, like all of you get to answer them in the same order as you presented. So I would start with the first one, which is when you were initially transitioning from academia to your alternative job sector, who did you turn to for advice, and what would you recommend graduate or postgraduate students to, like, who should they reach out to in order to find help in pursuing a different direction?
Yeah, definitely. I can give that answer. So when I was a postdoc at that time, when I was kind of, thinking of going to the job I'm currently in, I actually reached out to my grad school mentor. So I reached out to Josh Kuhn, and we had a really nice talk just, about the different career trajectories. He had seen people go to industry and he had known people there. So he had a good sense on that. But I also reached out to people who were in those types of positions. So, if you had former colleagues that had left academia and gone to different positions, or even if you've just met people at conferences and stuff, you can always reach out to them and just get a sense and ask them, hey, do you have like 10, 15, 20 minutes for a call just to get a sense of what it's like to be in a position that you're at, and that type of thing. So that's what I would suggest.
So if we turn to Lindsay, then what would your take on this be?
Yeah, if you're thinking about spinning out a company from research that you did when you were in academia at your current institution, a lot of your universities really around the world will have some entrepreneur or small business school, either separate within the university, or they might even have some sort of specific small business startup company spin out program. So it's called different things in different countries. So I would reach out to, if there's a business school or a law school in your university, they almost always will have someone to point you to, and they'll help you with kind of all the steps that you'll need to get started, and help you either find a co-founder or work with intellectual property protection, or the first steps for what do you need to do in your country to form that company, to make sure everything's all legally covered. So reach out to your institutions. They almost certainly have something to help you out.
Thank you so much.
Thank you very much. So thanks for the first question. So for the second question, this actually is a very...
We also have Maria as well today.
Oh, I'm so sorry.
Which I guess has quite a different outlook on it to turn into like the publishing area.
No, that's okay. Actually, my answer is similar to the answer that Chris gave. So I was also very fortunate to have a wonderful postdoc supervisor to whom I felt comfortable talking to. So she gave me great advice when I was exploring what I wanted to do. And she actually, when I mentioned that I would like to potentially apply for editor jobs, she encouraged me because she thought that it would fit to my skills. I mean, again, yeah, as Chris said, I would encourage everybody to talk to people, ask them to give you a glimpse into what the job is about. And I'm sure most people would be happy to spare the 10, 15 minutes to talk to you. And I would say, especially if you're undecided about what you want to do, keep an open mind. And you can talk to really anybody, even if it's a job that you feel like most likely would not suit you, it doesn't hurt to know what it is about and find out more, and then decide having more data at hand. And another thing you can do, of course, is, if possible, to apply maybe for an internship. That again might give you, internships are not offered everywhere. For example, we offer some, and I know companies do as well. And that may also give you an impression from the inside what the job is like, because again, sometimes it might sound nice when someone describes it, but you might not enjoy it when you get the chance to do it yourself.
Thank you very much. My apologies, Maria. So for the second question, this is actually one that is often a very important one, and a lot of us, transitioning from academia into industry, have this problem, and that is, given that graduate students often focus on a very niche area within a wider field, what would you recommend that they do to expand their opportunities within when leaving academia, and whether this actually represented a challenge, which I'm pretty sure it did at some point? So if you could start this off, Chris, that would be great.
Yeah, definitely. So if you're looking, if you've done kind of one area, within, say, proteomics, mass spectrometry, whatever you're doing. If you've only kind of focused on one area, that's not really that big of an issue if you're trying to leave, I'd say, academia, right? If you're trying to go to industry. The important things are kind of the skills you've learned. Generally those will have to be applicable to a job that you're going to seek. People will want to see that you have a core set of skills that can be applicable to whatever you're doing. It doesn't necessarily mean you have to have done the same protocols, workflows, or other things. It's just the skills you have can then lend themselves to learning whatever the specific techniques are that are going to be available or you're going to be using. So I think that's the first thing. You don't necessarily need to have exactly the experience to join, especially for us, right? What we're looking for when we're trying to recruit people is basically a core set of skills that we think can be translatable, and then also like the enthusiasm for the work that they want to do. Now, in terms of expanding your opportunities, one of the ways you can do that if you're a graduate student, there's lots of companies that have like internship programs. In those cases, you might be able to learn new skills and then also get a sense for what industry research is like. If you're beyond that, then I would really lean more towards, the networking aspect and going to conferences, talking to people from companies, understanding what their job is, what they look for in candidates and things like that. But yeah.
Yeah, because this can sometimes be like a double-edged sword, where sometimes when you do a PhD, it's so specific towards a field within a wider field. And when you start to apply for jobs, they don't necessarily want that specific. So I guess core skills is one of, the most important aspect.
Yeah, the core skills are important, and the demonstration of achievements and being able to finish projects, understand the particular goals of a project and being able to achieve those goals. Those are a lot of the things that are going to make you successful. If we have to train people, we don't assume that people are coming in are going to know the workflows the way we do them anyhow. So we end up, even if someone's done something similar, that they'll end up learning how we do it. So there's going to be some training anyhow.
Can we have your thoughts as well, Lindsay?
So Chris had kind of touched on this, and I think the number one thing you can do in whatever degree you're doing or whatever you need to do to get more opportunities is network. That is the number one thing you can do no matter what you want to do, whether it's go to industry or start a company or become an editor or any number of the other non-academic careers. Network, because you never know when the person you need to talk to 5 years from now is a person you just went and grabbed coffee with for 15 minutes. So go to conferences when you can, participate in the conferences. It is exhausting, but the whole point is actually, don't tell your advisors I said this, the whole point is to not actually go to the talks, but like to meet people, go around the poster sessions and be talking with people, make business cards, or make sure that you get their LinkedIn information, or however you're going to contact them in the future. Number one thing you can do is network.
And finally, Maria, do you want to add on to this?
Yeah, I mean, I agree with everything that was said already. Maybe something more specific in case some people are considering the editor path. There it is really especially important to have a broader view of science. So of course, yeah, going to conferences is something that definitely helps. Reading a lot of papers, maybe reviewing papers as well, if you have the option to do that together with your supervisor. And generally, because the main role of the editor, the main task is to anticipate what comes next. So to pick up upcoming fields, upcoming topics in a certain field. So that's something to keep in mind. Like, for example, if you go to a conference, even if it's on your field, try to see what's coming up based on what the outlook is that is given in the talks of the big speakers and what seems to be cooking, so to say, in that specific field.
But I mean, in your field, which is in the editing, if someone is heavy on the lab space rather than writing papers on the grant, would that cause some kind of hindrance onto his or her application?
No, so typically all editors are hired directly after postdoc. I mean, occasionally there are people who are hired directly after the PhD, but this is not that common. And what we really assess during the interview is the thought process when you are given a paper to read. So what really matters is whether you can evaluate with an editorial view, so to say, a manuscript. So to see, does it have potential? Does it fit to the scope of the journal? And it might be like, for example, when I started at MSB, I didn't have a particularly strong background. When you saw my CV on systems biology, it was only my postdoc that was 2 years where I touched a little bit on systems biology. So on paper, I would have been a very off candidate, let's say, for that job. But nevertheless, I applied and I did well at the interview and I was hired. So maybe something else to mention on that occasion is don't be afraid to apply for jobs even if you don't think you tick all the boxes. Because you never know, like Lindsay said, with the person that you have coffee at the break, you never know what's going to happen in 10 years. Maybe you work with them. The same applies to a job that might seem a little bit off for your CV, but it might be actually the job that you fit well. So
yeah. Thank you. Arshin, you want to?
Yeah, so I see we have some questions coming in the Q&A. So before we jump into those, I'll just ask a quick one. Were there any obvious trade-offs that you had to make when you decided to leave academia or during the actual process of leaving academia? Yeah, Chris.
Yeah, I can go. So the main thing that when I was deciding which way to go, one of the main concerns or thoughts that I was worried about was, will I still have some opportunities or openness to kind of, if I have a particular interest that I want to try, like a new method, a new way of doing an experiment, am I going to have the freedom, right? It's that freedom that you get in academia, and are you going to still have that in industry? So I was a little bit concerned about that. I mean, at the end of the day, the real difference there, right, is, in academia, you might have a little more freedom to kind of just try out things, spend more time maybe on stuff that may not have as big of an impact. In industry, right, a lot of times we have very specific focus on the goals, right? And a lot of it would be the goals of the company. Now, those can be quite broad, like Genentech, right? Our goal is to help patients, to develop therapeutics. So the things that we do every day, like we should always be keeping that in mind. So if we are thinking about even creating a new mass spec method or a new way of doing this analysis, the real question then becomes, okay, how do we believe that this would end up helping us reach those goals? How do we believe that this, and for us, that's really, how do we believe this new approach is going to help biologists answer a question that's going to help us make a decision on a particular therapeutic program? So in that case, it is maybe more limited, but what I found is that it can also be a little bit more interesting because it's no longer just like, what can we do, but it's really, what can we do that's going to have an impact? So, while the bounds might be a little tighter than it would be in academia, I think on the other side, it really helps you to focus on the activities that you're doing and make sure what you're doing every day is really kind of in the best interest of the ultimate goal, which for us is really that idea of like creating things to help patients. So that in that sense, it is different, right? And your mindset day to day might be a little bit different, but at the end of the day, a lot of what at least we do at Genentech feels a lot like you would in academia or like a research institute or something like that. So it doesn't feel too different, but again, there is always going to be that slight difference there.
Hey, thanks, Lindsay. I see you've been nodding to a lot of the things that Chris has been saying there.
So don't have a ton to add as far as
Yeah.
trade-offs. My biggest concern is like what Chris said, like, oh, I'm going to industry, therefore I'm not going to be able to do like cool academic kind of ivory tower research. And really that's not the case, in part because, like he said, in industry, the point is just you have an applied goal that you're working for. So you can make cool new methods. You can like mess around with a mass spec and like come up with these new proteomics methods. You just have to remember that the whole point is not just to do cool science for the sake of cool science. The point is, oh, well, now in a year, I can apply this to discover a new class of drugs or a new class of proteins, or help diagnose some different form of cancer. Like the whole point, just you have to think a year or two farther out rather than just, oh, I'm going to like work for a couple months on this cool new thing just for fun. You have to think, oh, but is there a point for what I'm doing? But that's really the only trade-off that I've had to make. We still have the highs and the lows of academia. We're writing papers, we are writing grants, like we are in the lab doing experiments. We have mentorship, we do like intern projects, we have younger scientists that we're teaching and growing their careers, preparing them for grad school, preparing them for whatever their next job is. So it's really honestly a lot like academia without some of the kind of like institutional support that academia has, where academia might have additional avenues or groups to help trainees. We don't have those because we're a very small company, or grant support, or additional kind of like logistic support. We don't have that, but that's really been the only major trade-off so far. We're still pretty little.
Okay, that's interesting that on both industry sides it's quite similar. So I guess it's cool now that we have Maria to chip in on the other element.
Yeah.
That could be, yeah.
Yeah, for me, I can't say, I mean, as I said, I was really pretty determined to leave academia, so I can't think of an obvious trade-off. So I was pretty happy with the choice that I made at the time. What concerned me a little bit in the beginning was whether I'm restricting myself in terms of choices that I would have later if I would decide that I don't want to be an editor after some point, because of course it's as an editor you acquire specific skills, and I was wondering whether that might be a bit too restricting and what could I do then afterwards. But thankfully I discovered quite early on from colleagues of mine who left and moved on to other jobs that it's actually, of course you cannot back, I mean you probably can, but it's very difficult to go back into research or to a more like a industry setup, but you can do many other things. So there is former colleagues of mine who are now scientific coordinators. They work as managers in funding agencies. Of course, you can move to different journals. You can freelance as a science writer and do many other things. So that was kind of reassuring to see, and that was my main worry, I have to say, when I switched.
Okay, cool. Yeah, so I think we'll jump into some of the Q&A. I see there's a few that came through. Said.
Sorry. Yeah, so the first question, Anuraj asked, and it's specifically directed to Pino, but feel free to join in from the panelists. So he basically asked about the journey that Lindsay took with her work. The question is, on the verge of finishing his PhD in computational proteomics, he's a little bit confused about whether to choose an academic or non-academic career, as people have suggested that research in non-academics is slightly oriented in a different way than in academics. So what factors did you consider when joining, I mean, in your case, when co-founding Talus Bio, Lindsay?
Yeah, I think Chris and I kind of answered the spirit of this question with the last one. As far as research being different in academic versus non-academic, I'd say it totally depends on the company. And even within a company, within that exact department or division that you're going to be working in, it can just vary wildly. If you're at a big enough company, different departments are just totally different vibes. So you just kind of want to check that out. I'd say at smaller companies, the vibe tends to be a lot more academic style of research because you're kind of the size of a lab. And as far as the factors I was considering, honestly, I just wanted to do cool science. I wanted to have my own lab. I had done the postdoc just because I wanted to do tenure track, and that's what you have to do tenure track, but clearly tenure track didn't work out for me. So this was a way for me to have my own lab, continue doing science I thought was important and interesting. And everything else was kind of like tacked on to the back of that primary factor. But Chris can probably talk more about what it's like to do academic versus non-academic science at a bigger company.
Yeah, and again, it will vary a lot between companies. So like Genentech has always been somewhere as a mix in between, like an academic institution and one that's more industry focused, because, we do focus a lot on publication. For most people's promotion track and job, as we're going through our job, we have to publish, right? We have to go out, we have to present, and most of us like that. And that's one of the things that draws people to Genentech. But that's not always the case, right? So there's going to be other companies where the focus may not be going to conference. Maybe you go to one conference a year or one conference every couple of years, or maybe you never really publish anything. You're just doing stuff internally, and you're focusing more on patents and other stuff. So, it's really going to depend on the company. And I think, and like Lindsay said, even the departments within companies. So if you're out there and you're interested in industry, that's one of the questions, right, that you have to kind of ask yourself is, what is important to you? What are you interested in? Are you interested more in going to, because there's some startups unlike Lindsay's that don't publish. So are you interested in going to a startup that's really just focused on achieving the next goal, right? Where you're really focused on developing a platform, finding a particular drug for a particular, disease area, because that can be very exciting, wearing many different hats. Do you want to go to a bigger company where it's a little bit more standardized and you're just kind of, kind of learning the processes, completing projects, working on really cool, interesting biological questions. But at the end of the day, maybe you're not really the one leading the publications there and stuff like that, or the projects at the end of the day. So there's lots of different areas. There's so many different companies out there nowadays who are hiring proteomics folks. So you're gonna have lots of different opportunities. So it's really just asking yourself, like, what are you interested in and what do you prioritize over other things?
And I guess if I could sort of like relate this question to Maria, how different was the writing element when you transitioned from academic to your current role in terms of writing the paper or editing the papers as opposed to writing it when you were doing your PhD?
Yeah, unfortunately, this is actually one of the parts of being in the lab that I miss, the writing, because now I don't get to write almost at all. We just read and evaluate and maybe sometimes edit a little bit. So yeah, we don't write that much, but that depends strongly on the type of editor job. So there are news editors, for example, they write more, or editors that handle more reviews in pieces like that. They get to write and edit a lot, but I unfortunately do not write that much. So I mostly read and write emails.
Yeah. Andreas, you wanna go for the next question?
So to follow up, we have a question that's specifically directed to you, Maria. What was the opportunity that got you to apply for the position as an editor? Do you just like see I don't know, a post about like the job application, or was there anything like specific that made you pursue the editorial path?
Mm-hmm. Yeah, so I mean, I have to say, when I was doing my PhD and in the beginning of my postdoc, I didn't even know that this is a career option, really. At the time, there was not as much information as now. There were this type of seminars like what we are doing now. They were not so common, but they were very few. So I attended one of them, and then I realized there was an editor presenting, and I realized that this sounds quite interesting and that might suit me. And I also realized that you don't need, I mean, I thought at the time that you might need some special training or something like that, which is not the case. So then I started looking for job ads and something opened up and I applied. So I applied for a few other editor jobs as well, where I was not invited for an interview. And as I said before, actually, this one was the one where I was least expecting to be invited. Most of the times these positions are, they are open, and you can see, yeah, most journals, they have on the website or more central job websites that you can find these jobs. But if it's something people are interested in and you happen to meet editors at conferences, you can always talk to them and they can keep you in mind, or write to them on LinkedIn. Like I received sometimes emails on LinkedIn, and if the people sound promising, then I always flag them to the person who is hiring in our office. So that's always good connections, as we said before, to have.
I guess like a key aspect here is to be really like curious about new science in general, I guess, as an editor.
Absolutely, yes.
Right.
Yeah, absolutely.
Irishan, you want to ask the next question?
Yeah, so is there anything that would make you go back to academia? Any reason you can think of, or any challenges that might come to mind if you were to go back, having made the transition successfully? Anyone, whoever wants to go for it first can.
I'm not sure. I mean, I never, you know, when I was leaving my postdoc, I was in the process of writing like a K99 and other things, and I've helped write grants before, but we don't have to do that at our company. I have to talk to people about budgets and stuff, but that alone would seem a little bit daunting. I don't know if I'd want to have to do that. So maybe if someone just gave me a bunch of money to do something, sure.
Okay.
Yeah, I'm not tempted to go back. Short answer.
I think I could be tempted, and the pretty obvious reason I might go back is if anything happens that my company goes under, or opposite way too. If my company gets acquired and they're gonna kind of turn it into something that's not what it is now, and it's not kind of fun anymore, I could totally see myself going back to academia to continue doing what got me into this alternate academic career, which is doing science. I think is cool and important. If I can do that in academia for whatever reason, I would totally keep doing that.
So
okay, cool. Yeah, Said, go ahead.
So we have another question. So we have a question from one of the audience. So she's asking, so all of the panelists went on from PhD and postdocs into their current positions. Do you recommend going into industry right after a master's degree? As I already know that I am not too keen on continuing in academia, but is a science job market open to, I guess, graduates after master's degree without a PhD? It's actually a good question. So if you guys had some takes on it.
Yeah, the industry is definitely open to that. So, you know, at the end of the day, wherever level you kind of ended up at really just kind of determines where you might fit into the particular like job family or a job family within a company, right? So companies will typically have particular tracks that folks might go on in their career. So if you came in with a master's, like there are jobs like that at many different types of companies. So you could definitely get in and then start working your way up. And the only difference if you went to get a PhD and then a postdoc is that you get slotted in somewhere else. So at Genentech, right, I did do PhD and a postdoc. By doing the postdoc and getting some papers there, I was able to come in a group leader position, where typically that's not something that happens if someone just has a master's. So it also depends a little bit on what you want to do. If your ultimate goal is to come in, and that's just at Genentech, but if your ultimate goal is to come in as a manager, basically, right, someone who's going to have a group and to kind of lead an intellectual area, then you kind of have to have that kind of two-step process, the PhD and the postdoc. But if you're going to come in and you want to work at the lab and then start off your career from there, then yeah, a master's is completely fine. And you'll see that, you know, master's students can actually, maybe Lindsay can speak more about the startups, can be very attractive to smaller companies, right? Yeah.
But in an extension to your discussion, can you still lead a group from a master's background, let's just say maybe after 10 or 15 years of experience, or does it necessarily need a PhD qualification to get there?
I'd say after enough, especially within, it depends on the company, like the track system they have, but I would say that with enough experience and demonstrating the kind of your abilities, for us is generally, again, right, demonstrating those like tangible outcomes, things like patents, papers, and other stuff like that, you can certainly be there. We have group leaders now at the company who've been there quite a long time, who they may not even have masters, right? Like you know, they've been at the company for like 20, 30 years. So yeah, that type of thing, I would say nothing is out of the question, especially for us at least, right? If you've accomplished the things that would be similar to someone who's accomplished that, it doesn't really matter that you didn't have the degree at the end, because if you've been working in the last 10 years, you've demonstrated the ability to do something as good as someone who just has a PhD, then yeah, you could do that.
Yeah, and Chris kind of alluded to this, but smaller startup companies might be more fast track than that, because if you're good and you fit what they need you to do, you can kind of rise really quickly, because as I kind of mentioned earlier, like titles are kind of meaningless at startups. They're kind of made up. So you could rise pretty fast in a smaller company if you had the skills that they were looking for.
Yeah, maybe to quickly say that for editor jobs, it's unfortunately that I know of, it's not possible to start only with a master's. So typically editors at minimum have a PhD. Most of them have also some postdoc experience. And actually there's also some people that are even more senior than that and have led groups for a few years, and then they decided to leave academia at this stage. So if for whatever reason that's something you are interested in from an early stage, it might be worth continuing and doing at least the PhD.
So in connection to the previous question, we have one that's like regarding to if you realize when starting off your postdoc that this is not really your cup of tea, like do you do better like leaving ASAP, or should you stick around and gather the experience from doing that postdoc, or what would your recommendations be in general here, as all of you actually pursued like a postdoc as well?
Maybe I can start, since I did a very short postdoc. Actually, I mean, as I said in the introduction, I realized very early during even at the end of my PhD that I didn't want to stay in academia, but I nevertheless decided to do a postdoc. That said, that was 10 years ago, so the times were a little bit different, and doing a postdoc was more standard. Now I know that the postdoc is viewed with a lot of skepticism. Not many people do a postdoc. for me, it was certainly beneficial. So I feel like even in these 2 years, my view of science matured a lot, even working in a different lab and being exposed to a different setup, different topics, different institute, all that contributes to your experience. My recommendation would be, once you realize that that's something you do not want to do, at least to start applying. So that is what I did. I can't say, of course, that what I did was the right strategy, but it worked for me without that I stayed and did a very long postdoc that was not leading really anywhere. But of course, it might be that from the time you start applying, it takes a while until you find a job. So probably the earlier you start applying, the better. But yeah, it's really difficult to recommend something specific. But try to, if you do a postdoc, try to make the most out of it in terms of learning, making connections, and really acquiring the skills that you feel are going to help you the most in what you want to do next. For example, if that is being an editor, try to write a lot, read a lot, review a lot, and acquire the skills that will help you for your applications later on.
Yeah. Do you, Chris or Lindsay, have anything to add in this? Would you consider like cutting it off?
Yeah. So if you had started, maybe it depends a little bit on how long you've been there. But one of the questions that would typically come up from someone, because we have interview committees and other things, like there would definitely be questions about like, oh, why are you leaving it before having something that came out of it? So that might be a challenge, right? Is that if you do leave before you had a paper or something else to kind of demonstrate that you had a project, you started it and you completed it. Now that may not be it may not be the end of it, but that would just be something you'd have to kind of be able to explain, like why that wasn't a great fit or why that situation wasn't working out for you. Because there might be some questions around that, like, okay, how are they going to view when they come into this position? So that would just be the one thing. We've definitely seen, there was some of that, especially around, there was some things like that, especially around the whole pandemic, right? Where people are kind of going some places for a short period of time, then switching out again. So if you are planning on doing that, right, that's just something to keep in mind.
Yeah, I would agree with everything that was said. And the only thing that I would add, as far as just strictly the experience of a postdoc, I don't think will be super meaningful for a lot of jobs. They won't really care that you just did a postdoc to do a postdoc. And as far as learning skills in a postdoc, I think most jobs aren't really going to care too much that you used an ACTA like fractionator or something like in your postdoc, right? So as far as learning skills, it's not like a great reason to do a postdoc or stay in a postdoc, or just the experience of having X years as a postdoc is not really going to be super meaningful. Like if you realize you don't want an academic career, a postdoc's not going to really train you to be better at industry. Whether that's editing, whether that's a government job, whether that's being a grants person at your funding agency in your company, like postdoc is not going to teach you to do that. You need to go do that job to do that. Postdoc's going to teach you to be a PI of a tenure track position, which is great. If that's not your goal, then postdoc is probably not what's going to get you to that next role.
Yeah, thank you so much. And we've actually heard that we're sort of running over time actually now. So I just want to thank all of you a lot for joining this session. And unfortunately, we'll have to wrap up here. But a massive virtual round of applause for all of the presenters and the entire discussion as well.
Yeah, thank you so much.
Thank you to all of the attendees as well for joining.
Yeah, and as Lindsay said, unfortunately some questions that we couldn't answer, and some of you raised your hands, but we couldn't really take your questions. Catch you at the next conference and take her for coffee, and you can definitely ask her your questions.